Welcome to our exploration of genetically modified organisms and gene therapy. Today, we'll dive into how society, environment, and health are interrelated and examine the implications and future impacts of these scientific advancements. By the end of this session, you'll understand the basics of GMOs and gene therapy, their benefits, risks, and controversies. Let's begin by setting the stage for why these topics matter in our modern world, especially as technology rapidly evolves and influences our daily lives. The relationship between society, environment, and health is complex and deeply intertwined. Advances in biotechnology, such as GMOs and gene therapy, have the potential to transform how we live, eat, and treat diseases. These innovations can improve food security, address public health issues, and even change the way we approach medical treatments. However, they also raise questions about ethics, ecological balance, and long-term safety. Understanding these connections helps us make informed decisions as individuals and as a society. Genetically modified organisms, or GMOs, are organisms whose genetic material has been altered in ways that do not occur naturally. This is achieved through modern biotechnology, allowing scientists to add, remove, or change genes to give organisms new characteristics. The process opens up possibilities for improving crops, creating medicines, and even developing new materials. GMOs represent a significant leap in our ability to shape the biological world, but also introduce new challenges and responsibilities. The foundation of genetic engineering began with the discovery of DNA in 1953 by James Watson and Francis Crick. This breakthrough revealed the structure and function of genetic material, paving the way for scientists to understand how genes carry information and determine an organism's characteristics. With this knowledge, researchers could begin to manipulate DNA, leading to the development of GMOs and gene therapy. The discovery of DNA marked the start of a new era in biology and medicine. 1973, Herbert Boyer and Stanley Cohen conducted the first successful genetic modification by combining genes from two different E. coli bacteria. This experiment demonstrated that it was possible to transfer genetic material between organisms, laying the groundwork for future GMO development. Their work showed that genes could be manipulated to create new traits, opening up a world of possibilities for agriculture, medicine, and industry. 1982, the Food and Drug Administration approved Humulin, the first GMO-produced medicine. Humulin is a type of insulin created using genetically engineered E. coli bacteria, making it available for diabetes treatment. This milestone demonstrated the practical benefits of GMOs in medicine, providing a reliable and safe source of insulin for millions of patients. It also highlighted the potential for biotechnology to revolutionize healthcare. Flavor Savor tomato, introduced to the U.S. market in 1994, was the first genetically modified tomato. By deactivating a gene responsible for fruit softening, scientists created a tomato that could ripen on the vine and still have a long shelf life. This innovation allowed tomatoes to develop their full flavor while reducing the need for artificial ripening. The Flavor Savor tomato demonstrated how genetic engineering could improve food quality and storage. 1995, the Environmental Protection Agency approved Bt potatoes and corn, as well as Roundup Ready soybeans. These crops were genetically modified to resist insects and tolerate herbicides, helping farmers achieve more consistent yields and reduce pesticide use. The approval of these GM crops marked a turning point in agriculture, offering new solutions to age-old farming challenges and sparking debates about safety and environmental impact. Golden rice was developed in the Philippines in 2000 to address vitamin deficiency, a major public health issue in many Asian countries. This genetically modified rice biosynthesizes beta-carotene, a precursor to vitamin A, in its edible parts. Golden rice was intended to help reduce health problems caused by vitamin deficiency, especially in regions where rice is a staple food. Despite its potential, golden rice remains controversial and has not been widely adopted by farmers. Golden rice is one of the most hotly debated GMOs in the world. While it was designed to reduce vitamin deficiency in poor areas, it has never been offered to farmers for planting. The controversy centers around concerns about safety, ecological impact, and the ethics of genetic modification. Golden rice illustrates the challenges of balancing scientific innovation with public acceptance and regulatory hurdles. GMOs have transformed the food industry by enabling crops to possess valuable traits such as drought tolerance, disease resistance, and improved nutritional content. These modifications help farmers produce more reliable yields and reduce losses caused by environmental factors. GMOs also offer the potential to create foods with enhanced health benefits, making them an important tool in addressing food security and nutrition challenges worldwide. GMOs play a crucial role in medicine, with insulin for diabetes being the first commercial healthcare product produced by genetically modified organisms. Beyond insulin, GMOs are used to produce other medicines such as growth hormone and vaccines. For example, hepatitis B vaccine is produced by genetically modified yeast. In the future, plants may be engineered to contain vaccines, offering new ways to deliver immunizations without injection. Genetically modified cotton has been developed to resist insect attacks, improving crop yield and reducing the need for pesticides. This innovation benefits farmers by lowering costs and increasing productivity, while also contributing to environmental sustainability. GM cotton demonstrates how genetic engineering can extend beyond food and medicine to impact other industries, such as textiles. GMOs offer several benefits, including reduced pesticide use, better protection against diseases and insects, higher crop yields, and potential cost savings for farmers. These advantages help ensure a more stable food supply and support agricultural sustainability. By improving crop resilience and productivity, GMOs contribute to addressing global food challenges and supporting farmers' livelihoods. Despite their benefits, GMOs also present risks and limitations. Insect-resistant plants may harm beneficial insects like bees, and herbicide-tolerant crops can lead to the development of superweeds. Farmers must pay technology fees for GM seeds, and patented crops restrict seed saving for breeding. Long-term health effects of GM food consumption remain uncertain, and ecological impacts are still being studied. Safety of GM products is tested in intense, short-term studies, but their long-term effects on health and the environment are not fully established. This uncertainty fuels ongoing debates about the risks and ethical considerations of GMOs. As new organisms are created through genetic engineering, ecological issues may arise, including potential imbalances in local ecosystems. These concerns highlight the need for careful regulation and ongoing research. GMOs can cause ecological imbalances similar to those created by exotic species. The introduction of genetically modified organisms into the environment may disrupt local ecosystems, affecting native plants and animals. These potential impacts underscore the importance of studying ecological processes and monitoring the effects of GMOs on biodiversity and environmental health. Gene therapy is a technique that uses genetic material to treat genetic disorders. By inserting healthy genes into cells, gene therapy aims to correct or replace defective DNA, offering long-term solutions for diseases that were previously untreatable. This approach represents a major advancement in medical science, providing hope for patients with genetic conditions and expanding the possibilities for personalized medicine. In 1972, Theodore Friedman and Richard Roblin proposed that people with genetic disorders could be treated by replacing defective DNA with healthy DNA. This idea laid the foundation for gene therapy, inspiring researchers to develop methods for delivering genetic material to cells. Over the following decades, scientists worked to refine these techniques and overcome challenges related to safety, effectiveness, and delivery. In 1985, Dr. W. French Anderson and Dr. Michael Blass demonstrated that cells from patients with adenosine deaminase (ADA) deficiency could be corrected in tissue. This breakthrough showed that gene therapy could be used to treat genetic diseases at the cellular level, paving the way for clinical research and future treatments. Their work provided proof of concept for gene therapy's potential in medicine. In 1990, the first approved gene therapy clinical research took place at the National Institutes of Health under Dr. Anderson's team. This milestone marked the beginning of gene therapy trials in humans, allowing researchers to test the safety and effectiveness of genetic treatments. The results of these early studies helped shape the development of gene therapy protocols and regulatory standards. In 1993, the first somatic treatment that produced a permanent genetic change was performed. This achievement demonstrated that gene therapy could create lasting effects in patients, offering new possibilities for treating chronic and inherited diseases. The ability to make permanent changes at the genetic level represents a significant advancement in medical science. In 2003, China approved Gendicine, the first commercial gene therapy product for the treatment of certain cancers. Gendicine's approval marked a major milestone in the application of gene therapy, demonstrating its potential to treat serious diseases. Since 2006, clinical successes have brought greater attention to gene therapy, although it remains an experimental technique in many countries. In 2016, the Committee for Medicinal Products for Human Use, CHMP, of the European Medicines Agency endorsed Strimvelis, a gene therapy treatment. The European Commission approved Strimvelis in June 2018, providing a new option for patients with genetic disorders. This endorsement highlights the growing acceptance and regulation of gene therapy in Europe, paving the way for future treatments. Gene therapy involves inserting a healthy gene into a carrier, called a vector, and transferring it to affected cells. This process can be performed inside or outside the body, depending on the treatment. The most common vectors are viruses that have been genetically modified to carry normal human DNA. Viruses are effective carriers because they naturally transport genetic material to human cells, making them ideal for gene therapy applications. There are several approaches to gene therapy, including replacing mutated genes with healthy copies, inactivating improperly functioning genes, and introducing new genes to help fight disease. Each method targets specific genetic disorders and aims to correct or mitigate the effects of faulty DNA. These approaches offer new possibilities for treating a wide range of conditions, from inherited diseases to cancer. Vectors are essential for delivering genes in gene therapy. The most common vectors are viruses, which have been genetically altered to carry normal human DNA. These viruses encapsulate and transport genes to human cells, enabling the correction of genetic disorders. The use of vectors is a key component of gene therapy, ensuring that genetic material reaches the targeted cells effectively and safely. Gene therapy offers several advantages, including the potential to treat genetic disorders with single or few administrations, improving quality of life and reducing the need for frequent physician visits. It also allows for targeted treatment of affected tissues, minimizing side effects and maximizing effectiveness. These benefits make gene therapy a promising option for patients with difficult-to-treat conditions. Gene therapy is primarily an experimental technology and is highly regulated to maximize patient safety. Potential risks include unwanted immune reactions and the formation of tumors. The effects of current gene therapy approaches are limited to the treated patient's cells, and modified genes are not passed on to future generations. Careful monitoring and regulation are essential to ensure the safety and effectiveness of gene therapy treatments. While gene therapy holds great promise, it also faces limitations. The technology is still experimental, and not all genetic disorders can be treated with current methods. Risks such as immune reactions and unintended genetic changes must be carefully managed. Ongoing research is needed to improve the safety, effectiveness, and accessibility of gene therapy for a wider range of patients. Genetic engineering, including GMOs and gene therapy, raises significant ethical concerns. Some argue that it is unethical for humans to alter and engineer organisms, while others support the potential benefits for health and agriculture. These debates reflect broader questions about the role of science in society and the boundaries of human intervention in nature. Genetic engineering poses challenges in agriculture, including ecological impacts and the need to study agricultural production systems. GMOs can affect soil health, biodiversity, and crop management practices. Understanding these challenges is essential for developing sustainable agricultural strategies and ensuring that genetic engineering supports, rather than harms, the environment. Further research is needed to understand the ecological processes involved in agricultural production systems. Studying the effects of GMOs on ecosystems, soil, and water resources helps identify potential risks and develop strategies to mitigate negative impacts. Ongoing ecological research is crucial for balancing innovation with environmental protection. Clinical experiments are essential for advancing gene therapy and understanding its functional mechanisms. These studies help outline predictive approaches, patient-related outcomes, and upcoming challenges in gene therapy development. Rigorous clinical research ensures that new treatments are safe, effective, and accessible to those who need them. Future perspectives in gene therapy and GMOs depend on continued research, clinical trials, and public engagement. Addressing existing problems and challenges requires collaboration between scientists, policymakers, and communities. By studying functional mechanisms and predictive approaches, we can develop safer and more effective genetic engineering technologies for the future. Societal implications of GMOs and gene therapy are far-reaching. These technologies can improve health, food security, and quality of life, but also raise questions about access, equity, and regulation. Understanding the social impact of genetic engineering helps guide responsible innovation and ensures that benefits are shared fairly across communities. Environmental implications of GMOs and gene therapy include potential impacts on biodiversity, ecosystem balance, and sustainability. Careful management and monitoring are necessary to minimize negative effects and protect natural resources. Environmental considerations must be integrated into the development and regulation of genetic engineering technologies. Health implications of GMOs and gene therapy include improved treatments for genetic disorders, enhanced nutrition, and new medical options. However, concerns about long-term safety and unintended consequences remain. Ongoing research and regulation are essential to ensure that health benefits outweigh risks and that new technologies are safe for patients and consumers. Public debate about GMOs and gene therapy is ongoing, with strong opinions on both sides. Supporters emphasize the benefits for health and agriculture, while critics raise concerns about safety, ethics, and environmental impact. Open dialogue and transparent communication are essential for building trust and guiding responsible decision-making. Regulation and policy play a critical role in the development and use of GMOs and gene therapy. Governments and agencies establish standards for safety, effectiveness, and ethical conduct. Effective regulation ensures that new technologies are used responsibly and that risks are minimized. Policy decisions must balance innovation with public health and environmental protection. Research and development drive progress in GMOs and gene therapy. Scientists work to improve techniques, address challenges, and discover new applications. Continued investment in research is essential for advancing genetic engineering and ensuring that innovations are safe, effective, and beneficial for society. Education and awareness are vital for understanding GMOs and gene therapy. Informing the public about the science, benefits, and risks helps build trust and encourages responsible use. Educational initiatives can empower individuals to make informed decisions and participate in meaningful discussions about genetic engineering. The global impact of GMOs and gene therapy extends across health, agriculture, and environmental sectors. These technologies have the potential to address worldwide challenges such as food security, disease treatment, and sustainability. International collaboration and regulation are necessary to ensure that benefits are shared and risks are managed globally. Future challenges in GMOs and gene therapy include addressing ethical concerns, improving safety, and ensuring equitable access. Continued research, regulation, and public engagement are needed to overcome these challenges and realize the full potential of genetic engineering. By working together, we can create a future where science benefits everyone. Summary: genetically modified organisms and gene therapy represent powerful tools in science and medicine, offering new solutions for health, agriculture, and environmental challenges. While these technologies bring significant benefits, they also raise important questions about ethics, safety, and ecological impact. Ongoing research, regulation, and public dialogue are essential for guiding responsible innovation and ensuring that genetic engineering serves the greater good.